So it's my great pleasure to introduce Dr. Stephen Friend. Dr. Friend is an authority in the fields of genetic resilience, cancer biology, and digital health. In his early work at Dana-Farber and MIT, his team cloned the first human cancer susceptibility gene. And then while on the faculty at Harvard, he was jointly recruited with Lee Hartwell to co-found the Seattle project at the Hutch. From there, he co-founded and led Rosetta Informatics, where they developed some of the earliest RNA expression approaches to assess the aggressiveness of breast cancers. It's fair to say these results have been enormously impactful for breast cancer treatment and survivability to this day. Shortly after this work, Merck acquired Rosetta, and Dr. Friend became the senior vice president for oncology, where he rebuilt the cancer franchise using molecular profiling techniques, some of which would later show value in assessing the sensitivity of tumors to Keytruda, to treat melanoma, lung cancer, head cancers, and other cancers. After working at Apple from 2014 to 2016 on digital health, he is now co-founder and president of 4U&Me, the founder and chairman of Sage Bio Networks. In addition, he is currently based at Oxford University as a visiting professor of connected medicine. Stephen is both a fellow, both an Ashok and an AAAS fellow. Today's presentation will be on boundaries between unknown and known unknowns for individual symptoms from genomics to wearables. Thank you very much, Dr. Friend.
Thank you very much, Mike. Let me see if I can share my screen. And on sharing the screen, let's see if I can put it onto play. And Mike, can you confirm that you're getting a signal from the machine?
Yes, that looks great.
Great.
Yes.
Yeah, this is cool. I can close my eyes and look back like 24 years. And since wandering through posters at Cold Spring Harbor Lab meetings, trying to figure, do I need to go to that session? And trying to absorb. So it's a new way to do that, but it's bringing back memories, and it's not about PTSD. Usually speakers limit a talk to, they say to tell you, only have 3 or 4 takeaway messages. So today I'm going to try one. So what is it? The lesson that all the themes going to go around is that it's essential to make insights, to use ways to simplify, and then hard to live with the forgotten consequences. So let me say it again. It's essential to make insights, to use ways to simplify, and then hard to live with the forgotten consequences. So this address today is going to be around that theme all the way through. So let's start with some examples of such simplifications that could give insights. Let's go back a quarter of a century to the studies that Lee Hartwell and the group had done in the Seattle project at the Hutch. So this was the schema that we thought for gaining insights for molecular alterations given this. So this is pre-having the human genome sequenced, et cetera. So the idea is go into tumors, find their molecular defects, identify analogous defects in genetically tractable organisms like yeast or C. elegans or flies, and then alter the analogous gene representing those primary defects, then perform synthetic lethal perform synthetic lethal screens to identify secondary targets, then come up into analogous secondary targets in mammalian cells, et cetera. That obviously is a simplification. And the type of evidence that something like this could be helpful is this work that Philippe and Chris Roberts did, which is they went into yeast, they modeled in these genes that if you're working in oncology, you'll recognize are analogs of defects that exist in human tumors, and built out those strains. And in this instance, they would apply a drug like cisplatin or mitoxantrone and ask whether in that yeast readout, it could give a clue as to mechanism of action. Now, the second part of that description is, of course, as you know, although that works in that well-defined simple story, or in that simple system, the ability to extrapolate and do that in mammalian cells has been a multi-million-dollar industry in many places, and occasionally that works. So let's go to a second example. Second example is the work that Tim Hughes did when we were at Rosetta with a number of others. We joined a consortium run by Ron Davis, who was looking at yeast, non-essential genes in yeast and disrupting those, and looking to see whether we could build a compendium and use that as a query, as a way to query for lots of things. And as you probably know, you can do this bidirectional clustering, where you cluster by profile and you can cluster by transcript response. And we were able to show that when you took a compound like FK506, its signature would basically be triangulated, and you could see how similar a compound was to a particular target, for example, the histone deacetylases. And so that's an example, again, where that concept of building a compendium, and even the more powerful ones that have been built out in yeast and some other organism, it begins to become really difficult to use as the complexity enters the system to where you'd really wish you could be using it. The third example is now, well, let's go into human cells. Let's take RNA expression in human cells. Let's take one type, for example, in breast cancer. And in this, teaming up with Laura Vantvier and the group at the NKI, taking thousands of patient samples, could you come in and narrow it down into a subset of, in this instance, 70 genes that were able to sort of parse for how aggressive a woman's breast cancer was. And it's a mixed end here. So in that instance, using mammalian gene expression profiles in breast cancer, that actually has turned into a tool that allows many women who are looking at how aggressive their breast cancer is to have a good clue of whether they should not even get certain therapies or they should get other therapies. But the zinger there is, so that if it worked there, could it work in other tumors? And for the most part, again, that has been a disappointment as you extrapolate it. Try to take a finding in one place and extrapolate it out to others. So these three examples, sort of in the omics realm, about the difficulty of extrapolation, a lot of it, we feel, comes down to inappropriately assuming that the conditions that you have are generalizable, and the problem in terms of unknown unknowns. And with that in mind, if you're ready, I'm going to go off and try to tackle, look at how we're trying to tackle a real hard problem. So with that concept of beware, you can get insights, but they may not extrapolate. I want to go into a project that we've been working on for basically 8 years. This is a description of a problem. It's not the actual one, but the contour here, lines here, and the valleys and the hills are meant to represent a hypothetical set of energy minimum, or another way of representing shifting from one state to another. And what we have been trying to frame, I wouldn't say even solve yet, but try to frame is, what would it take to be able in health to look at what it takes to shift from one disease state to another? What would you need to have, and what should you work on if you're intending on doing just that? And what we woke up to quite quickly was that this would require a shift in where you were collecting information, how you were gathering that data, and the timing for that data to be gathered. And what we hoped was that if you could take the intermittent assessments that go into medical records that are done in clinician's office that say this person now has this disease, didn't have that disease. If there was a way to take the various of our 5 senses that a clinician uses in examining a patient and find out a way where some aspects of touch and hearing and sight, maybe not taste or smell, but if you could find a way to capture those outside a clinician's office, that then you might be able to have the layers and the depth and the dimensions of data that might allow you to look at those disease transitions. And at the same time, funded by the Robert Wood Johnson Foundation, we realized that in order to collect that type of data, that you would need to have studies that actually treated people in different ways, that you would want to have studies where they were participant-centered, where someone who was had some various thing that you were together wanting to follow was willing to give their data and work with you. So this concept of data, a lot of data, and a different value proposition for what it was like to be in a study, not a subject, but a co-owner or participant. And we realized that the devices that could give us this semi-ubiquitous data could make a lot of sense to look to using a smartphone and other wearable devices. And so by about 5 years ago, we got into where it's some semblance of the types of studies that might be able to be done. We had a great relationship and linkage with Apple, who was interested in trying to come up with different ways that individuals who are wearing their devices might be able to participate in research, and help put together this program called Research Kit that would allow individuals to enter a study independent of what institution they were at, join a study, e-consent, and begin to get that type of high dimensional frequent data that I was talking about. So here are the first 5 Research Kit apps that came out. They were built as a framework for clinical studies. They had these various modules for consents and surveys and tasks and passive data. And the key idea was to have open code that you could put up on GitHub. You could have the data, you could have the open code, and people would sort of potentially build an ecosystem of working together. I will say that in some respects, this has flourished remarkably. I don't know whether it's millions of people, but it's certainly hundreds of thousands of people who have been in various studies using this research kit, and it's analog in Android. And yet the concept of building an ecosystem where people were willing to use other groups' tools, and the concept of working on other people's data fell short because of human nature. And I think the nature that universities set up where they want their own proprietary way that someone will use a tool that is quote unquote giving recognition to that institution. It's a topic I'm not going to go down any further. But let me tell you briefly about Empower, which was one of those. Here are some of the individuals, in particular, Andrew Trister, Ray Dorsey, and Arno Klein, that helped put this effort together. This project had over 20,000 participants. So you know, normally a Parkinson's disease study might have dozens of people, and this had hundreds of people with the disease and more out of that large number. And what they were able to do is they could log into the app with secure password. They would 2, perform activity, 3, all that data encrypted on the phone would 4, be sent to Bridge. That process data could be sent back to the phone. 5, that personal identifiable information could be separated. And then that data 6 could be transferred to Synapse, which was part of the tools that both Bridge server and Synapse that Sage built out. And then that would allow the data analysts at other places to do with appropriate credentials to access that coded study data. And then it could be available for researchers that met the governance qualified researcher threshold that was set up and used a lot at Sage. So these slides are going to be available, so you can go into it later, but you can imagine there were various components in the app. There were data feeds, there were features that could be used that were collected off those, in those, you know, more than 10,000 individuals. And two take-home lessons here. One is when you had not a single score for a disease, which was usually in the charts called the Universal Parkinson Disease Rating Scale, UPDRS. So when you don't have a single way of measuring, but you could say, let's look at the aspect of the disease captured by taps or gait speed or balance or voice frequency. What's interesting is if you rank order, let's say 30 individuals for their ability to do taps, when you go and you look at gait speed or balance or voice frequency, you'll see that actually each person's disease is not going down some linear path, but in fact is occurring in a way that should not be aggregated if you're trying to look at causation. Second thing that was a take-home message is there are many differences in individuals, but the intra-individual diversity was something we could never have seen if you couldn't look every day. So this is 4 months, 120 days, looking every day, pre or post someone taking their medicine. So a single individual over 4 months. And what you can see is they're having good days and bad days. And some of those days are ones where they're getting a benefit from the drug or not. This is really valuable, as you can guess, if you're trying to look at whether one drug is working better than another instead of having those block terms captured every several months. And that has become something that several pharmaceutical companies have gone on to take advantage of. So it's that level of detail that you can get out of that simplifying insight. And since then, as you'll recognize, there are tools like the Apple Watch that are beginning to bring those back to individuals in gentle, informative ways around their health. The papers that came out on this type of data showed more and more that the clinicians assess, this is my version of this, showed that clinicians' assessments can be good, but in terms of objective data, the data that could come in from machines and looking exactly off the sensors can be extraordinarily accurate. The other point I want to make before I move on is that these large-scale longitudinal high-resolution data sets to follow a symptom take advantage, and this work that was done by Evidation and Apple and Lilly, to not, this is important, not have a single data stream that's coming in, but pull in data, whether it's from the phone and the watch, or the digital assessment apps, or a bedded app, or apps that are on the phone. And this phrase, multimodal data stream, was able to be shown quite beneficial when you wanted to study something as abstract as cognition. And in doing that, it's those multiple data streams that are asking, one pattern from another. So I think sometimes we'll hear people naively say, well, what is the way I want to measure X or Y as a symptom? And of course, what we come back with is right now, deeper levels of data are going to be more helpful. So back to this concept of what would it take to be able to follow symptom transitions at an individual level. So you're saying, how could I forecast a change in someone's medical conditions? In 2017, I left Apple to work on this concept of individual symptom trajectories, formed a non-profit that has been working on the fundamental unknowns of using smartphones and wearables, and how, this is even harder, how to effectively return it to individuals wishing to navigate with health and disease, where all the data, findings, algorithms, and apps are put in the public domain. So for you and me, has been doing a series of studies trying to figure out where can we follow individual symptom transitions, and how do you give it back to people? How do you give them agency and context to do that? The model that we have used is to have participatory strategic funders, and then instead of having a large set of people at one location, this was actually pre-COVID that it made sense. Now, of course, it makes sense. Have leading experts that are at existing institutions and leave them there and have contracts with whether it's universities or companies or startups or nonprofits, and have them reach out to them and say, who's got the best cohorts, who's got the best assessments, who's got the best devices and data flows. And so this has been a whole series of what we call coalition partner alliances to do this work. And this is where you'll see many of those, and it's not all of our partners, instead of on the last slide. So I want to do a shout out to all the groups that have been helping us. And we've been using all-day sensing. As I said, we sort of moved from just using the ring, or sorry, the phone, but also the watch and ring and other devices that people have made. You can guess the types of things we follow. And what we've been doing is taking sets of devices, looking at the sensors on them, building out ways where we would have sufficient data feeds to be able to look at something like cognition or mood or sleep or gait or fatigue. So for the last 1.5, 2 years, as we've gotten going, the projects that we've been doing are around two things. It's been detection and forecasting symptom transitions, like I said. And then as you'll see near the end, looking at the consequences of stress and lifestyle changes impacting those symptom transitions in chronic disease. So those two areas. Now what I'm going to do is I'm going to zip into examples of some of the actual studies on forecasting symptoms, and then later some of the examples of looking at the effects of stress. So in the area of symptom transitions, working in oncology, recognizing that there are liquid biopsies, there are scans that can be done, et cetera, but usually those are pretty expensive. We wondered if you could sort of build some sort of like early warning detection signal for tumors that have these studies are called hero studies because it's about helping enable real-time observations. And to do that, what we did was went to situations where there were aggressive tumors that were hidden, that were hard to find, and where they were aggressiveness that we could have a certain expectation, unfortunately, of having new tumors arise. So these studies around aggressive glioblastomas, looking at pancreatic tumors, and now beginning to think of working in ovarian, are studies that are about to launch in the next couple of weeks. The process at Hopkins and at Princess Margaret are coming along. And the concept there, you can imagine, is we'll have devices on individuals after they have been treated, had their surgery, whatever. And in that situation where the potential for relapse is going on and sadly high, we will be looking until someone has a relapse and then going and looking at all the data feeds that we had to ask were there any common themes that might have been suggestive for doing that type of work in this, again, defined pilot zone, simplified zone, and then figure out how might we be able to build it from there. Second example is working in pregnancy. We have a large study called Better Understanding Metamorphosis of Pregnancy, BUMP. This is to follow 1,000 women from very early in their pregnancy all the way through 3 months post their delivery. In that study, look for particular symptoms like emesis, fatigue, edema, mood, etc. And the pilot is in a fun way either going to start today or on Monday. So as soon as I get off this symposium dialogue, I'm going to go check to see whether we can enroll that first patient. So as I said, we took 8 symptoms and we're going to look at those and see how can passive signals, active tasks, and surveys allow us to have a way of following symptoms outside a clinician's office. It's a virtual study. The goal is really, in some ways, though it's very large, it's also a pilot, because I think the things we learn in this would allow us how to do a larger one. But we want to confirm the functionality of integrating this digital and clinical data We want to look at whether the digital signals associated with predicting the pre-specified symptoms are possible. How different are they in the different symptoms? We want to look at the intra and inter-individual variations in terms of the strengths of those signals. And we're hoping to identify routines in those people's lives that help us give better assessments and the power of using those routines. So what we did to find those 8 symptoms was we went through all the symptoms, or at least a very large set of symptoms in pregnancy. And we chose ones where we thought our sensors were best suited to pull out data. So this is a spider chart. So this is just showing for fatigue in gold, for cognition in blue, for emesis in red, some of the key sensor data that we're going to pull, but it shows that we could have done many more. We chose to focus on some where we thought our likelihood of getting good signal was a little better. These individuals will have, in addition to the phone and all the things that are on their phone, they'll have a Garmin watch on that now also has aspects of SpO2 and respiratory rate. They'll be able to get off that. The Oura Ring that we've been using in many of our studies, it's particularly good at nighttime and sleep and HRV. And then you may not be aware of a company that has a scale that don't just get your weight, but because of the way they have put the electronics together and what is on that scale and how it can work both as an ECG and conductance, they can get ejection fractions and pulse arrival times and a number of things, BMI, that we think will be really helpful to have among these individuals as they go through their pregnancy and then postpartum. To give you a sense of the scale and the scope of this, you can see that on those devices, the Ring, the scale, and the Garmin, we have a number of data feeds we'll have off them. In the more virtual world and how people are interacting with others, there are opt-ins and opt-outs to using Google Takeout, RealID, Facebook, Instagram, and then a whole set of active tasks that are on the study app. And then there'll be study surveys and active tasks and bi-weekly check-ins with engagement experts that we think will give us insights and some unstructured data. Let's see, sorry. Just a last slide on this set on BUMP. This is the amount of data that we have coming in sometimes every minute, sometimes every hour for a year on 1,000 individuals. So we're hoping that, yes, this is a data set that's about understanding pregnancy, but we're hoping that there'll be, whether it's headaches or other things that are going to be happening, that we may learn a lot about the sort of the progress of symptoms and disease, not just about pregnancy. The concept is break the rule that you can't go deep and wide at the same time on human data. Now for some sort of fun and sneak preview. Mackenzie Wildman, the lead digital analyst on our programs at Evidation, showed up one day, and it wasn't quite this complete, but the way she told the team, by the way, I am pregnant, was to have us guess what was this. And here is an example. So this is just obviously a single person looking at changes in sleep efficiency and sleep score. But now let's go to resting heart rate. This is obviously maternal heart rate and heart rate variability. So I think there's going to be real data that we're going to get out of that BUMP study. In terms of the assessments for virtually all of our studies, the team at Vector, particularly in Anna Goldenberg's lab and in Marcia Gashimi's group that's also up there, have been designated as our alliance partners for looking at the data. I put in some examples for those who are interested in this symposium on the types of work they're doing, and I think you'll see why we chose them, plus the fact that I think they have been thinking hard about large amounts of data in a clinical setting in a way. That said, all of our data will be put over on the Sage Synapse system, and we're hoping that anyone in the world who's interested in problems will be in a position to have the data, the findings, the algorithms, the apps to be able to use it. I'll skip this on trajectory clustering that they're doing and move to the other types of studies that are swirling in our heads. And that is looking at how stress affects end organs. And so what I don't mean is how does stress affect the risk of suicide and the mind. What we're interested in is when one is stressed, whether it's by physical or mental stress, various reasons to be stressed. There are things that happen in that body, in our bodies, that trigger pretty blunt responses in the body, with sympathetic and cortisol levels, etc., sort of going, okay, I'm in a new state, that then have intermediate consequences, changes in sleep and mood, cognition, and at the same time end up, we think, giving end organ deterioration. So in that context, you can probably guess what we've done, which is the same way that we were using wearable devices, surveys, active tasks to study symptoms in chronic diseases. We sort of turned that, pivoted that turret over to look at how could we collect much deeper, richer, more frequent streams of data related to stress. So getting engineering signals from smart rings and watches, scales and body patches, right? Sounds familiar. And then the smartphone passively being able to do the same things we were doing with symptoms, but now for stress. And also having active tasks that we ask people to do, in this instance, active tasks that allow us to assess immediate and intermediate stress. One of the projects that has not yet started, I bring up because I wish it would get started, which also means I wish it would get funded so we could get it started, is to do an in-depth assessment of stress response in a very defined way, a narrow window, going from state A to B to A. And this support study is one that says, let's take cancer patients who are in remission and need on a periodic basis to come in and get assessed as to whether they stay in remission, and to have ways where we can follow those individuals' stress levels and responses before that, and then in that 2-week interval leading into that assessment, during the assessment, and afterwards. And we think that that should give us some granular differences, ABA, to begin to mine some of the aspects of support a little deeper that we might not pick up. So I think aspects of voice and how your voice is that you know changes when you're stressed. I think we'll do a better job of figuring out what is that stress look like by having that ABA, to facial patterns, to skin conductance, etc., to go, hey, what is a good way to track stress in other situations? We have 3 studies, 2 of them ongoing, the Crohn's and diabetes, one of them which is getting ready to go up in Toronto on Li-Fraumeni syndrome, where we're asking questions about, can we forecast relapses or flares in Crohn's disease by following individuals, not their symptoms, but now we're looking at, is there evidence that we can show objectively that the level of stress in people is different, and that is coupled to flares. One real quick comment, we just wrote up something where we looked at Crohn's patients, not with wearables, and looked at COVID-19 and asked how much did COVID-19 affect the disease, and where were the sort of causes, or at least the associations. And as you can guess, stress went way up, and was in terms of how people felt. And there were, like many chronic diseases over the last 8 months, changes there. I'm not going to go in from the standpoint of time and go into the Lee-Fraumeni study, but if anyone wants to ask me about that later. On diabetes, we've been looking at how stress produces glucose excursions in individuals. You may know that up until last decade, most people who had diabetes got insulin. That was considered the treatment. Then people have begun to come up with ways of treating the pancreas itself, not supplementing insulin, but actually affecting how the body is reacting as it becomes more and more in a diabetic state. To projects where people have said, well, let's change people's eating habits. But I think what we're interested in looking at is, could wearables give us a clue of the behavioral excursions that lead to the obvious food and exercise excursions that lead to the glucose excursions, and that maybe what individuals in the next years might be able to work more on is there a way to have a feedback loop with themselves about what is going on and the causes of the things that produce the behavioral excursions, that produce the glucose excursions. So it's a project up with a group in Sweden, Anders. I think the last study that I will go through is one that was partially funded by the Gates Foundation this year, when a lot of work shut down in terms of many of the studies we were trying to get started. We couldn't on wearables. We looked at the COVID-19 frontline healthcare workers. Ended up, we thought we'd have 250 participants, but we went closer to 400 who had an Oura ring on them. So they were following, we were following certain sensor data from them. They had apps on their phone, they had questionnaires, and they had weekly, bi-weekly links with engagement experts who were looking at what was happening to them. And our goal was to ask, could we see when individuals were having one or two shifts in a row, what was happening to how they were becoming overwhelmed, those immediate and intermediate measures of stress, and did they have enough time to adjust, to compensate, to get back to baseline? Were we giving individuals a chance to recover? So stress and recovery was the concept of that study. In the next couple of weeks, we'll have the last participants out. It gave us a lot of data, and we added a subarm where we, several subarms, some of them looking at cortisol, some of them looking at interventions. But the most interesting one was there were enough individuals that could wear the Garmin watch as a subarm to look at what was happening both on shift and off shift, and the timing and stress. So I think that's going to be a data set that, as we finish and put it out there, may be of interest to individuals. Specifically looking at the data from this was again done thanks to McKinsey at Evidation. They do an awesome job of processing and aligning the data from the various data streams to make what they call a behaviogram that is useful for many purposes. One of the obvious important ones is to look at whether there are data feeds that are missing or something is odd. So it is a way of looking at curation and more to have those. And in terms of specific data, so again, a sneak preview, if we looked at individuals who rated themselves as very calm or stressed, we could go in and look at given individuals on various nights, and the data signal for whether or not someone was or was not stressed, as you can see, was rather powerful. So again, thanks Mackenzie for that. Now I want to turn for the end into what's part of that theme that I was trying to stay on, which is those unknown unknowns, those unknown complexities, and what are we thinking they might be that are going to prevent us from extrapolating. So what is it that we should be taking into account so that we don't solve for particulars and have a better sense of things that will work in a more general sense. Here in this quadrant presentation by Stephen Gray, one way for people who are working in machine learning is to look at those situations where there are simple cause and effect relationships, complicated, where with sufficient time and resources, you can get to those cause and effect relationships, called complicated. Complex, the term where the order is not apparent, we can't understand cause and effect, and chaotic, as you can guess, is can't do it at all. And what we've been trying to understand is what is where in the process of being able to solve some of these healthcare issues with wearables, and what does it take to move a complex to a complicated, for example. So in this instance, this idea of looking to whether you can extend or generalize or search in red, and the knowledge that's held by us, we're looking for risks where they're seen elsewhere, risks where we can see where they're emerging, looking at potential causes, looking at insights from benchmarking, and using that to anticipate what it is that may be problematic. So I'm going to give you 3 examples of what we deem to be potentially solvable issues that, until we sort them out, are going to crush our ability to use these wearables. The first, which actually has nothing to do with wearables per se, is about taxonomies of disease. So today, when you go and you try to get a cause or an association, whether it's GWAS or anything else, our disease categories that we use are like, this is someone who's depressed or has Alzheimer's or has arthritis or cirrhosis. And from various projects that we're doing, we're convinced that until there's a better taxonomy, until there's a better differentiation into substates, that any times where you're trying to do, is this associated with this, or predicting depression or anything like that, if we use those almost 19th century bins, I didn't say garbage cans, bins of all these patients who have these 5 of these 10 symptoms, we're going to say they have the same thing. Until we figure out a way to rebuild some of those taxonomies of disease, it's going to be really hard to do forecasting for individuals. Secondly, there is I think close to pathetic, that's the right word, yeah, it is, assumption that when someone goes through a disease, they are going to go down some linear path from prodrome to early to moderate to advanced. What I mean by that is almost every textbook has a progression of disease that is more talking about the level of the disease and not whether there are different paths. And so as we saw with Parkinson's disease, and you will see with, we think, any disease, chronic disease you're following, until you develop the tools that allow you to look at whether that's singular or branching, and be able to have that branching structure, similar to what I think ahead of almost all other disease groups, those who are looking at evolving mutations in cancers can do. But imagine that same type of question about other chronic diseases is going to be important to shift out of that pathetic assumption that it's a linear progression for most diseases. Not everyone is going down that same pathway structure. To the third one, which is something that we feel as if we've been caught in a lot. Almost always when you put in a grant or a proposal, or you say you're going to work on something, you say, I'm going to work on sleep and, or I'm going to work on cognition and, or mood and, et cetera. And when you look at a singular sort of cause consequence zone, you'll get an answer, but it would be much different if you actually were looking more broadly at the overlapping causes and consequences. And so I think we're thinking, what does it take to be able to untangle that and actually look at causation? So with the high resolution data that we have, having timing, having ordering, having some of these possible micro rhythms, I think one of the things we're interested in, anyone who wants to work with us on it, we'd be glad to go beyond association and talk about causation. And what is it, really what we're interested in is, what's the experiment that we could do that might allow us to look at some aspect of causation, because we have enough data with regard to ordering and micro rhythms and timing that might help. And then finally, from our work on stress and recovery, where these healthcare workers gave so much of themselves, they in fact will be co-authors on the hundreds will be co-authors on the paper that we're going to put out. We learned so how important it is to have people in such studies be felt as co-owners and real participants. And we don't think we've done enough yet to work on that. We've been thinking of like what is needed to be able to have an ecosystem where it's like by each other, for each other, where people are willing to share that data. And how could you build communities where there, yes, in this era, how could you build communities where there is enough trust and knowledge and support and privacy to be able to do that? There's a project in 2021 we're hoping to begin to pilot in this area around looking at women who are going through menopause, who want to work together and help have those roles in that community, and look at passive and engineering signals and do the same types of things that I've mentioned before. And then I want to go to Shoshana Zuboff, if you don't know her work. The other concern that we have, so I've had concerns that are about thematic and theoretical, but practical. So Shoshana Zuboff at Harvard has done a lot of work on looking at the digital century and what's happened to the promises that had been there about what it would feel like to live in a connected world with all this digital information. And I think she's been focusing more on tech giants and seizing control of information and of learning itself. One book that is a little thick, but it's I think well worth it. It's called The Age of Surveillance Capitalism, where she looks at where she is looking at many things. We have been using those themes to look at the navigation of chronic illnesses in that age of surveillance capitalism, on who's going to actually have the data, who will have the knowledge, how will the individuals gain advice, what's going to be like for others to forecast future states of health. And along those lines, been working with the CRE and with a number of companies, Sage, to build out an open hardware, open source, open firmware device so that we could do studies all the way through where the way the data was collected, where it was kept, where who used it and everything like that, would have access to all the sensor data and be inexpensive and be used for these broad projects. So that's the end. I've tried to present some things that may help us going forward. And the theme of the talk has been, it is essential to make insights, it's essential to make insights to use ways to simplify, and hard to live with the forgotten consequences. In turn, the expected phases of excitement from early on seeing something to realizing how hard it is to extrapolate. So thanks very much.
Stephen, thank you so much for a wonderful presentation. So let me take the privilege as being the moderator to ask kind of the first questions, but feel free for others on the call, feel free to ask in the Q&A box. So let me ask sort of one science question and one sort of meta question. So I'll start with the meta question. So on the call today, there's a number of kind of students and postdocs that are sort of wondering about their own career trajectories. I get asked this all the time. And given your kind of lifelong experience, being incredibly successful both in academia and in industry, I'm wondering if you have any kind of advice or sort of how you can think about the trade-offs between these environments, things like, I don't know, resources, funding, independence. And then ultimately, I think everyone here just wants to have impact on the world. And, I'm really in awe of how much impact you've had. I wonder if you have any kind of advice to everyone on the call today about how we can make sure we're most impactful with the time we have.
That's a good, hard question to start with. In terms of impact, I think, so maybe I can make 2 points. I think that it is dangerous to get on a roller coaster through academia, plotting what is the next expected badge to have, and how do I get those badges? Not because that's in the person at all. I actually think almost every scientist would say that is not what drives me, and I wouldn't do it. But I do look at particularly academia as having these sort of medieval ladders of now you're a lord, now you're a baron, and it's almost like very hard to resist doing something that you think will be a short-term recognition. So I think that the advice on the impact is the people that I have seen who have been able to have an impact that they didn't expect are ones who had some passion, crazy idea, and looked more at how do I do it than whether or not others think that it's doable and it's short term. Just a small aside, Mike Gilman and I who was at Goldspring Harbor Lab for a while before Biogen, were in the lab, and he spent his like 5-7 years on his PhD doing extraordinarily, almost impossible experiments, which came together as a thesis. Yeah. And a lot of people went, whoa, Mike, what are you going to do? How's this going to go? He had in his head, he knew what he was trying to solve for and to work on. So I think it's an encouragement to those who are early in their career to be looking, what do I really want, and taking a little bit of a longer arc on it, and then going wherever it is that allows you to work on that. So my moves around and different ways of being in different jobs has not been that I got bored or wasn't interested. It was that I was always looking for, am I in the right place to do this particular part that I will need to get to the next? So assuming that really the focus should be a real soul searching on longer term, and am I in the right place to actually get to that, or am I just convenient and so going down the roller coaster?
I really like that sentiment about, if you kind of focus on the big picture and then just go wherever is going to support you the best, that you'll always be successful. And then if I can, let me follow up with a kind of a more scientific question. So you kind of enumerated 3 major challenges about, rebuilding the taxonomies, sort of studying the disease transitions, and then untangling cause and consequences. I like to think with enough data, we could rebuild the taxonomies in just in a very data-driven fashion. But I'm wondering about your thoughts on, especially how we're going to conquer, the fact that there could be a branching path to how the disease transitions may unfold. In the limit, if everyone is going to unfold in a sort of a unique way, how are we ever going to find any commonalities that we can actually address?
Yeah, that's really good. I agree with the way you've put one in one category, one in the other. I think that we are so early, and I think that's where I like thinking of a century ahead of us, is that we are so early in doing what to do with this awesome high dimensional data. And to your point, we don't know whether they're going to be thick branches or thin twigs, and are there going to be scenarios where, depending upon what you're looking at, actually the same quote disease may actually have different dimensional structures for other. And so I think that the project we're doing on menopause, I think, is the one that if we can get up to, in the thousands, might tell us whether the archetype structure of individuals who are going through menopause is fundamentally different. And that's why no one has figured out what causes hot flashes, or no one has figured out, what's setting off aspects of sleep. So I have no idea is the answer. And it's the question to ask, right? So when I talk to people like Mihaela van der Schaar or others and go, what's really going to mess us up? I think we don't know it's going to be thin twigs or thick branches. And so that's a really important experiment. And I'm sorry to harp on this, but I think finding things, and I think there are a dozen of these things which, if we knew, would help others. One of the things that we have dreamed of putting up is a wall of questions, not a wall of answers, but a wall of questions around wearables and chronic health that sort of have a Stack Overflow nature. And you look for getting your project up high on that list and hoping that many people will help you on it. So that's a different way of doing science. But I think that that problem that's there is one I would throw up and say, put that on the board, see if people will work on it.
I see. I guess we'll never know until we start collecting data, just how many paths there can be.
Yeah.
Well, I see there's a bunch of questions in the Q&A box. So let me kind of zip ahead here. So this one comes from Simon Lee. Thanks for a great talk. At the end, you touched on who has access to the data. Do you have any ideas on what society can do to reap the benefits of this technology without, for example, people being unfairly penalized by insurers?
Yeah, I'm really worried about this. And why I say that is, if you knew how much data, I think people talk about like the 5,000 bits of data that every night people extract from any individual on an average, right, in the virtual world. If you knew, or people realized, how much more accurate the sensor data is that's coming off you in the real world, it suggests that being able to forecast people's behaviors, and it's not just health behaviors, but forecast what is someone acting like, what they're going to do, I think is an enormous concern. And so I think we have to either figure out ways to keep some of the aspects of the data being processed on the devices, which would help, or having it such where there are exclusion clauses which say, I do not, I'm willing to enter this study, but it cannot be used for X or Y. So I think it is about insurance, but I see it not just about insurance. I see it for a lot of things where people might be penalized.
I guess the concern is, do we think those guarantees will be strong enough? And I guess especially in this area, it seems like legislation can be rewritten and things can be used in unexpected ways.
I was thinking whether I was going to wander onto the election, but I think we had assumed that everyone's vote would count, and that the rules about how it would be counted would not fall apart because someone had decided they wanted to run things different. I don't think that's that far away from the worries about the fact that you may say there's an exclusion rule and then what happens with it. So sorry to bring up the election.
Okay, moving on. So next question from Leonard Williams. Thank you for the great talk. Would you be able to point me to more information concerning the projects you mentioned working on Crohn's disease management?
Sure. There are two ways to do that. One is you can, anyone who wants can email me. I think it's in the Slack, friend@foryouandme.org. I'll get to it over in a couple of weeks or so, not tomorrow. But if you go on the website for You and Me, there's a study description on there that is a good placeholder. So on the site, and within the next 2 weeks, we're going to put the full grant proposal for everyone that we can on the website, because and the protocol, because I think most academic sites don't put their sort of their funded studies and protocols up. So we're going to put those on the website. And so in about a month, you can get to the protocol, you can get to the particular, what was the proposal, and that should help.
And then I noticed in your talk, you mentioned the Synapse platform at Sage. Seems like there's a lot of good data there as well.
And more coming. Yeah, they've done a remarkable job.
Great. Next question from Kenneth Ho. Thanks for the very interesting talk. I wonder about the noisy big data that you're likely to collect from wearable devices and to generalize those data to solve more specific personal illnesses. Isn't it a bit like having weather balloons collecting all sorts of data all over the world, but it'd be very hard to predict specific weather at a specific time and place using these noisy data?
Yeah. So the answer is yes. And a lot like it. And I like the analogy. And I've worked with some of the people in, so when you go to, using knowns to query unknowns, and you look at large data sets, the people in weather, as Kenneth, you may know, were working on this stuff like in the 70s. And the difference between looking at a hurricane and looking at a micro cyclone and a tornado is very close to what you're talking about. I think that right now, what we're hoping to do is to show that it is possible to forecast individual symptoms in some individuals in some diseases in order to figure out how hard is it going to be. That's where we are, and I don't know where that is on the weather analogy, but like we're way back, way back. Even though it seems like we have a lot of data.
Okay, so we have to start someplace, even if it's noisy today. Ziyang says, can you elaborate more on if it's possible how the device monitor stress and lifestyle?
On if it's possible and how wearable can monitor. Yeah, I think that it will be possible to have a way of letting someone know that objectively they're being overwhelmed. And let me give you an example. From the stress and recovery data, stuff that we're going to write up, we had people telling us whether they felt they were or were not stressed, so subjective measures of stress. And we also had objective measures of stress. And what was very interesting is those were discordant. They were significantly discordant. So there were people who would report, and more importantly, I think they're reporting to themselves, not to us. But I think one way of some people coping is go, I'm fine, I'm not stressed, okay? And what we're really interested in knowing is what is it, subjective or objective stress, that's ending up hurting the end organ? So if we end up finding that actually objective is not subjective stress, and objective stress is what is putting then I think people would like to have something that's similar to looking at your heart rate when you're running and going, I'm going too high. I would like to think that next 5 years, it would be possible to have wearable devices that look at sort of the equivalent of how overwhelmed you are, and is that coming back down or not? And I think that could be helpful.
Yeah, I agree. And plus, I mean, I think the sensors could be very exquisite. So I don't know if I would recognize if my heart rate was off by 5% or 10%, I might just not even notice it. But I think it can pick up these much more subtler indicators before you could be conscious of them.
Yeah.
And then the final question from Alan Min, could you comment on how in these projects you dealt with missing data? For example, what if someone wanted to wear the watch but not give certain other data types?
Yeah, that's good. One thing I'm impressed with is that each of these questions are ones that we worry about and think about. And this one here, I'm going to use it to make a point that I missed making. Usually when you have wearable studies, particularly if there's a high amount of friction, 1 to 2 weeks after you start, there's a significant classical drop-off in wearables of something that's as much as 80% in people who are really have become connected to it, and that missing data becomes significant. So there's a general problem with missing data. What we've been doing, and I recommend where you can, is these participant-driven, co-owner, reaching out to individuals on a biweekly basis approach that we've been using has allowed retention rates and use of device, and someone not being a parole officer, but saying, hey, you know, I notice you're not using your watch. Why is it? And getting them back on to doing it. And so I know you asked, you know, on how in these projects do you deal with missing data? Sometimes you just, as you can guess, you have to take that out because there's nothing. Sometimes you can do some extrapolation, but I hope it helped to emphasize something I'd forgotten, which was the importance of building studies where people are not subjects, but they're co-owners and they're interested in what's going on as a mechanism to fight the aspects of poor retention of people in wearable studies.
That's a great sentiment about let's build a community, let's make everyone involved, and then that will make them so much more engaged and having any, you know, I get reminders on my watch, but you know, you quickly learn to ignore many of those.
Sure.
But if we can build community, then you'll be really engaged. So Stephen, thank you so much for an excellent keynote presentation. We really enjoyed that. We really appreciate you sharing your words of wisdom. It's really exciting to think about what the future will hold with all these new data coming in and the integration of data that we can do in the future. So thank you very much.
To everybody. Yeah, right. Bye.
And then we're going to take a short break here.